another community invited, which is CCP-QC for quantum computing. And indeed, so we already have a superposition of states in a way. So today's talk will be given by Sabrina Maniscalo, and we are delighted really that she accepted to talk to us about quantum computers for life sciences, why, how, and when. Sabrina is a professor of quantum information computing and logic at the University of Helsinki. She's also a co-founder of a company called Algorithmiq with a Q at the end, and you can find the website. So Sabrina has been working on quantum technology for over 20 years, focusing on noise in quantum devices, complex quantum system, and quantum simulations. And today, I think our intention is to give a broad introduction to quantum computing, but also with the perspective on application in biology and business model. So I think that all communities should find something in there. So Sabrina, the floor is yours, and thanks again for accepting to talk to us.
Thank you very much, Nicholas and Sarah, for the invitation to speak about what we are doing at Algorithmiq. And I will now start sharing my slides. And yes, here. Yes. Soon you will see. Do you see?
Yes, we can see. Yes, I can see the slides.
Perfect. And as anticipated, the topic or the title of my presentation is Quantum Computers for Life Sciences, Why, How, and When. I will talk specifically with respect to Algorithmiq, that is our quantum software company. We are focusing on algorithms, and specifically on algorithms that already are applicable to near-term quantum computers. And we are a company which is based in Helsinki, in Finland, so the headquarters are in Finland, but we also have offices in the UK and in Ireland. And we are incorporated also in the States. So we are about 40 plus employees. So we are growing. We recently closed our Series A of fundraising. And we are working towards proving quantum advantage first for chemistry and then for the life sciences. But what I will be talking about here has to do also with what is called the quantum utility, and it is precisely a moment in which we will see that quantum computers prove to be useful for certain applications that could be more scientific, more academic, and then eventually also commercially relevant. So what I will do, another observation before I start here, is that I decided to give really an overview, because I know that the audience is mixed, without giving any technicalities really about neither the algorithms nor the approaches, but to have a general idea of what we think are interesting problems in the life sciences that can be tackled using quantum computers. And I will highlight what we think is the key enabler to scale up quantum computers simulations from proof of principle to useful and commercially relevant use cases. And of course, I will also try to describe the business model of Algorithmiq, and in particular, our product that is a platform called Aurora. So these are much more obviously in a limited amount of time, which means that clearly I will have to, my choice has been to give more a panoramic view rather than an in-depth view. And this will allow, I think, for those of you who are interested to contact later on and to follow up with discussions that may be depending more on your specific interest in the field, whether it is quantum computing or algorithms or applications to life sciences. So this is to set the ground. Well, the why, how, and when are the three questions we want to ask for. And I will begin with the why, and the why of our choice of a specific vertical, that is the life sciences, arises from the R&D challenge that pharma companies have been facing in the last years. Indeed, it has been reported that while the spend is up 10 times since the 1980s, and they're talking about the annual R&D spend, the number of new molecular entities that are brought to markets over the year remains approximately the same. So it fluctuates, but remains the same. And at the same time, it is also known that approved drugs very often lack precision. This is a way of illustrating this fact with this infographic. For every individual they help, recent investigations show that the 10 highest grossing drugs in the US fail to help between 3 and 24 individuals. This is, of course, something that really calls for an improvement. And the first question is really, what is the reason for this? And I think that it is probably widespread, the feeling that actually the reason of this is an oversimplification of the relevant cell biology when it comes to the modeling and understanding of how drugs act inside the human body. So which is our perspective, so algorithmic perspective on how to tackle this problem with the help of quantum computers combined with machine learning and AI techniques. Our framework is called, we named it quantum network medicine, and it is a multi-scale approach to tackle the complexity of the cell biology. It's a multi-scale approach because consists of different parts. Indeed, here I illustrate 3 of these parts. The first one is really a data-driven identification of the relevant network biology. So this first part here is not run on quantum computers. So it's classical or quantum-inspired. And this is part of a platform that we have developed that is called Aurora, and specifically is Aurora Network Medicine. So it's a sub-part of the platform that is focused on this type of problems. And again, the goal of this part is really to identify really which proteins in our body are safe targets with therapeutics effect, and which instead can be hazardous targets, because, for example, could lead to toxicities. And this identification is done by using network theory approaches or machine learning approaches or combined approaches, starting from data sets, omic data sets. I will describe this part in the following. Then we have a second part, which is not really proprietary. It's the part in which, once we identify a number of proteins from protein-protein interaction networks, for example, we need to model the protein structure in order to identify, for example, relevant pockets where the drugs bind to the protein pockets. And this it's actually something we are not developing in-house. So we are using AlphaFold, machine learning methods, crystallographic data, whatever is existing already. But it's not something where we exploit in any way the power of quantum computers. In the future, there are a number of interesting proposals to actually use quantum computers for solving problems like protein folding and so on. We think this is something extremely interesting, but more, let's say, for a fault-tolerant, for a future era of quantum computers rather than near term. And finally, we have the third part, which is part of Aurora chemistry. And this is really where quantum computers really shine. These are ab initio molecular simulations that can be run on quantum computers to describe, for example, bindings with high precision binding of small molecules to protein pockets. Now, why this last part is particularly suitable for quantum computers? Now, this is an oversimplified from some points of view perspective, but it captures some main principle. And it has to do with the fact that eventually molecules are quantum systems, and in certain cases and situations, they need to be described fully quantum mechanically. And when you need to describe an object of a complexity like coffee or even penicillin at fully quantum mechanical level, level, you quickly run out of memory on classical computers. Of course, we all know that for penicillin, it's not necessary to have a full quantum mechanical description. There are many effective methods that allow to classically or semi-classically describe the properties that are necessary to really to take into account the relevant information for certain complex molecules. But we will identify during the talk which situation and which use cases are actually more interesting from a quantum simulation perspective. So eventually, to simplify in a nutshell, really the message, there are instances in which simulating molecules requires quantum computers, and therefore, really in a very clear or slogan message, this specifically is a quantum problem that needs a quantum solution, that needs quantum computers that possess similar properties as the properties of molecules or any, if you want, electronic structure problem type of simulations. And because they possess this property that could be entanglement or superposition, they are better suited for a simulation of electronic structure problems. So this is the background in very simple terms. And then what I will argue in the following, or I will present in the following, are where in the drug discovery pipeline we think that quantum computers can really be useful, and where we can prove quantum utility in the next years. And specifically, I will talk about target identification, where we use methods that are quantum inspired, so use this quantum network medicine approach. And this is, of course, to better identify potential drug targets. Then I will discuss about lead identification, and here I will present some ideas on alchemical Hamiltonian methods that are allowing to efficiently explore the chemical compound space. And finally, lead optimization. And particularly, I will focus on lead optimization. Well, our interest is in mostly on covalently binding drugs and photoactivated drugs. But for the sake of time, I will focus on photoactivated drugs examples in the next as an example in the presentation. Now, instead of diving into the how, I will make first an attempt to elaborate on our perspective on the when, okay? And in order to understand the when, it is important to distinguish between two different, although not completely different, but in a way, time different, maybe in terms of times, modalities of computation. On the one hand, we have the near-term quantum computers. These are quantum computers using so-called physical qubits. Quantum bits are the unit of information, of quantum information. And these are devices that have size of hundreds, 50, hundreds, maybe thousands of quantum bits. And I'm talking about providers, hardware providers from different companies that are already working with these numbers. For example, IBM has been recently deploying a 433 qubits device. There is the announcement in their roadmap of more than 1,000 qubit device. Why the size, although the size of the quantum computers, so the number of qubits involved in the quantum computer is obviously not the only important metric, but it is connected to the complexity of the problems that one can tackle. So to the number of, to the complexity of the molecular simulation problem that one can tackle. So in this sense, it is important. But of course, it's important to remember that together with the size of the simulation, what is very important to take in mind are factors or metrics that have to do with the somehow precisions, or better, the resistance to errors of quantum devices. And this is really partly an engineering problem, but also with the speed of the quantum computation, because very often it is claimed that quantum computers are much faster than classical computers. And of course, certainly for today's quantum computer, this isn't true. I mean, they are actually rather slow, and they are very prone to errors. So near-term quantum computers are what we have today, and the type of algorithms that we need to use on these devices is what we call the hybrid algorithms. So these are algorithms that are not only quantum, of course, they have a quantum part because they run on quantum machines, but they also have a classical component. So in this sense, they are a hybrid. And when I say classical component, what I mean really is a number of techniques and methods that are used in nowadays, for example, in medicinal chemistry, computational chemistry. So I'm talking about the DFT method, the MRG method. So conventional methods to describe electronic structure problems, molecular dynamics, and so on and so forth. So you need to think of them as really working together synergetically in near-term quantum computers. So what will happen in the future, and this future is uncertain, really. Here I have a question mark with 2033, but of course it can be unknown whether it's before, whether it's later, is that we are progressively moving towards technologies that will allow to incorporate error correction methods that allow basically these quantum computers to work in a fault-tolerant manner. So in a way that they are not affected by errors, since the errors are corrected along the way. Error corrections methods are actually methodologies that are used also in conventional computers, but in quantum computers present certain difficulties that are typical of the devices themselves. So we are moving towards quantum error-corrected and fault-tolerant computations, but we are not yet there. And in particular, there are very important challenges that are engineering challenging that are needed in order to reach fault-tolerant quantum computers. We need to drastically reduce the noise and imperfections in the chips, and we need also to scale them from a few qubits to millions of them. So this is an effort which is currently ongoing, and that basically all players in the quantum space, in the quantum computing space, are in a way or another involved with. Now, when it comes to the current roadblocks, so if you think instead, everything I will talk about is focusing on the near term. So I'm going to focus first on what we can do in the next 2, 3, 4 years before reaching fault tolerance. Okay, this is where we are addressing our interest for now, which means obviously that as we move towards fault tolerance, many of the algorithms, of course, will keep being useful, and the reason will be clear later on. Actually, it would require some technical discussions, but and I will do it if you are interested. Now, currently, with near-term quantum computers, there are important roadblocks to the so-called quantum advantage, so to the demonstration that quantum computers can actually do things that classical computers cannot. And these roadblocks have to do with the interface between quantum and classical computers. It's really non-trivial to find a smoothness and seamless interface between the two. And generally, these classical computers are really HPC computers. There are issues with initializing quantum computers, in certain states that are useful for the algorithmic development or, if you want, simulations of electronic structure problems. And also, most importantly, it is important already now, and especially now, to figure out how to efficiently and scalably mitigate errors, not correct them completely, but mitigate them. And there are a number of approaches to do so. And specifically, when it comes to algorithmics, I will show 3 key ingredients of what we do that allow to tackle these 3 main roadblocks, and therefore shorten the time that will be needed to quantum advantage. I will skip this. So now, next question is how? How do we plan to do this? And specifically in Algorithmiq, we have three main components of solutions that allow to scale up, as I said before, the use cases. The first one has to do with mapping interesting problem to quantum circuits. I talked before about quantum bits or qubits, the fundamental units of quantum information, and quantum circuits are what are manipulating in the device really these qubits in order to perform certain simulations at the level really of combination of gates. And so how do we map an electronic structure problem into a series of gates and circuits that are run on a quantum computer? What we are doing specifically in chemistry application is we have devised way to significantly reduce the circuit depth via hardware-tailored transpilation and measurement efficient ground state finding. So this is a technical way to say we need to compact as much as possible and optimize as much as possible the mapping of the initial chemistry problem into the quantum computer in order to, in a way, make it sure that with the limited capability we have today, we will be able to run simulations that are significant or useful. And then the second part is our own framework. We extract information on the quantum computer via readout process that are proprietary. So we have devised a way of extracting information from the quantum computer, which allows for a number of improvements when one calculates properties in chemistry, for example, several RDMs. And finally, the most important part, and I will just not go into the details of these three parts because they're very technical, but the last part is what we call as a post-processing, post-processing mitigation noise, so noise mitigation approach. So this is where we use HPC to remove errors and noise from the calculations. So let me go in a bit in more detail in how our platform looks like, how Aurora Stack looks like. So at the bottom really of this figure here, you see the quantum hardware. So we are not quantum hardware providers. So we use quantum hardwares of different implementation, different physical platforms. But specifically, we are working a lot currently with IBM. They are our partners, and we are using their devices, although our algorithms are agnostic to the specific physical platform, but they are optimized to run on certain physical implementation. And on top of the hardware, there are some, we call them primitives really, that allow to extract information on the quantum computers, but also to remove the noise. And these are proprietary Algorithmiq's let's say a part of the software. Now, what is important to notice is that these pieces are not only just useful for the chemistry applications and also healthcare and life sciences, but they are also applicable to other verticals. So they can be used, for example, to simulate materials, new materials, or other problems that are not in the chemistry and life sciences framework. And then we have chemistry applications. Here, our main partners, we work with Cleveland Clinic as part of a Wellcome Leap project that is aimed at demonstrating in the next 3-4 years quantum advantage for chemistry applications. And also we collaborate with AstraZeneca. And the healthcare and life sciences instead are collaborations with Harvard Medical School and Lawrence Livermore National Laboratory. So just again, to briefly recap what I was saying before, the way in which the chemistry component works is that one inputs a certain quantum chemistry problem, the input is pre-processed using HPC cluster, then that part of the problem which is impossible to do with conventional methods is run on a quantum processing unit, and then the outcome needs to be again post-processed on the cluster in order to remove all the errors due to the imperfections of the existing devices. And finally, the result is provided as the output. Now, At this point, I go a bit more in detail on the applications, and the applications that I will describe. I will start with the target identification applications, and in particular, therefore, with quantum network medicine. So again, we are on this side of the quantum network medicine part. So this is the data-driven identification of the relevant network biology. So in this case, our starting point or our starting analysis are really bioneural graphs. And in particular, we work with data that are related to proteins, to diseases, and to drugs. So different data sets that we combine to understand better information on the on potential, for example, drug repurposing problems, just to mention one. And this needs to, so the very first thing that one needs to notice is that when one looks at data sets like the protein-protein interaction network, so the human interactome, and we here this is an example of one of the PPIs. Generally, in this specific example, for example, there are about 9,000 proteins and 64,000 interactions that are indicated by these links. But it is well known, and the link obviously between two proteins are when the two proteins are known to interact. And the important point here is that this is estimated to represent only 2 to 11% of the network. So this means this is really, in a way, problematic because one needs to complete this data set. So the problem of completing these graphs is very relevant. And therefore, one of the approaches that we have investigated is to use so-called limb prediction methods that are well known and very well used in network theory in order to estimate the probability of connections between proteins which are missing from this data. This part of the analysis is based on a concept is called quantum walk that is used very much actually in quantum information theory for certain algorithms. For example, search algorithms, some search algorithms use quantum walks. But in this case, without going into details, what we do is to use a quantum walk method in order to calculate probabilities for a walker to move from a certain node of the networks to others, so diffusing across the data set. And in doing so, it sort of maps or understands or tests the existing structures of the network. And in this way, eventually, this allows us to predict links with certain accuracy. And again, I'm happy to talk more if you have questions. So this will not go into the details. I will just say, however, that link prediction is not something obviously that we have discovered. It has been since the very beginning of network theory extensively used, and this is in particular, it's particularly interesting, or it has been particularly used for networks that are more social networks. But these are very different with respect to protein-protein interaction networks, and therefore the question of how and in which way we can adapt link prediction methods to PPIs is of interest. And here I have, as example, two different types of data sets, two different data sets. And I will show very briefly a results that is summarized in one of our publications here, in which we compare the quantum walk algorithms with other algorithms that are state-of-the-art in the literature for link prediction. And we see that the quantum walk algorithms is generally performing better with respect to the others. The line that you see, the blue line, exactly, the higher is the blue line, the higher is the performance of the algorithms in predicting missing links. And this basically, of course, in general, one has to do extensive study across many data sets, and this is also what we have been doing. I will show it in the next slide. So actually, it seems it is very promising. This is not run on quantum computers. It's a quantum method run on classical computers. We have used the same approach for a problem known as disease gene prioritization, and here the main point is to find new targets given a known disease gene. And also in this case, the method is based on random walks, similarly to the link prediction. And in these slides, which contains way too many results, what we are doing is just, so again, once again, the message is the blue link is the quantum method compared to others. And we are going across very many different data sets to show consistency. So if you see, if you change the data sets, the blue line remains approximately higher than the other methods, which shows that actually the method is consistent as we change the data sets. And finally, I will not talk about drug-target interactions networks, but I will just mention that these techniques are also applicable, and we have applied them to drug-target interaction networks, and in particular, these are particularly useful for drug repurposing. Now, this ends the part related to the quantum-inspired methods, and also the methods that are used to give context to the cell biology and identify potential targets. Subsequent discussion goes to lead identification. So which type of methods can we use in order to understand or to identify new leads? And these are alchemical Hamiltonian approaches. And again, here the baseline will be more on quantum simulations. So simulations on quantum computers, quantum processing units. Okay, so generally, the type of framework we are considering is the framework in which we have certain variational functional group around that fixed scaffold. Here we have the example of two ligands, and we compare these. So initially one considers this in a gas phase, then passes to aqueous phase, and then within a protein complex. What is generally considered is the free energy difference that is used in order to rank potential ligands. And of course, the main idea is to explore the functional group variations. And the problem that very often is faced is that when one uses molecular dynamics, even when complemented with DFT methods, for example, this seems to be, there are a number of problems in scaling up the complexity. Now, what has been done, and in particular, I'm referring to this publication, is that a proof of principle example of an alchemical Hamiltonian that allows really, in some way, to explore the chemical compound space without limitations to the known scaffold structures. This has been presented and tested on a quantum computer. Again, it's a very, very simple proof of concept example. The problem is that the current implementations very likely will produce unstable or even non-synthesizable molecules. And therefore, our approach is to use geometry optimization in combination with AI-driven additional cost functions in order to limit the search space to something which is stable, so to a subspace, if you want, that is stable, the space of stable and synthesizable compounds. And of course, one can add other AI-driven cost functions which could take into account, for example, pharmacokinetic factors. What is the main message here? Okay, the main message is that you have a certain part of the problem that can be run on quantum computers. And then you have, however, the fact that you will need to complement this with a number of approaches and techniques, in this case, AI-driven methods that will allow us to really input a variety of complex factors that are not captured by the quantum simulation per se, by the ab initio quantum simulation per se. And these are results where we benchmark our algorithms with the one of this paper, and I will stop it here. I will not go into the details. Final part, we focus now on the lead optimization case. For the lead optimization case, as I said, we find particularly interesting the case of photoactivated drugs. And in particular, for photoactivated drugs, of course, have been used since long time for cancer treatment. Indeed, there are examples of early cancer drugs that are very effective in killing cancer cells, but that they have the counter side effect of course, being very non-specific in binding to biomolecules. And these side effects can be minimized if the drug is selectively activated in the tumor sites. And this can be done via photoactivation. Basically, this is something which is well known. This article summarizes the techniques that have been known and studied. But of course, it is also well known that of course that this is computationally challenging. The reason why this is computationally challenging is that it does requires not only the knowledge of the ground state, but also the excited state knowledge. And excited state chemistry is demanding computationally. This project is something that we work in the framework of the Quantum for Bio Wellcome Leap Foundation. And I will just cite 2 examples of simulations which are not yet at the quantum advantage level, but that we are currently working on. And the first one is for BodyPi. That is, again, it's something which is, in a way, it's a very well-known case study in this field and in this framework. And what we are doing for simulating in this case, for us, it's important to develop the sets of algorithms, the algorithmic suites of methods that allow to study excited state chemistry. And what we are doing precisely is to do so by means of informationally complete measurement and approaches. So again, this is in combination as a benchmark of DFT and TDDFT in particular for calculating the properties of the molecules. And another example while the previous example is something which is commercially utilized in clinical trials, either commercialized or even used in clinical trials, the second example is psoralens, also well known, and now we are talking about radiation therapy. The previous case was UV light. This one is radiation therapy. The dynamics of the mechanisms are different, but of course, once again, the leitmotif, the main problem remains the same from the chemistry perspective, because you need to describe the electronic structure of both ground and excited state, but most importantly, one needs to understand the dynamics of the energy dissipation pathways, and this is crucial in order to understand really the reactive oxygen species generation. And again, in this case, this is the type of algorithms that are currently under development, and for which we will start testing in 1 year from now. However, just to give an idea of the complexity of the photochemistry for photoactivated drugs, this is a diagram that describes potential relaxation mechanisms. And it is known that one of the main difficulties really is in calculating conical intersections, as well as, for example, singlet triplet crossing. So what's the idea behind? The idea behind is that one of the crucial for us way to proceed in order to understand or to bridge the gap between what end users or researchers are trying to do and what quantum computers can do nowadays is to have and to work in collaboration, in our case, with the researchers at Cleveland Clinic, use their data, use the best classical algorithms that they are using, and try to see if quantum computers provide advantage in these specific examples. And again, so the problems come from there, and the solutions that we try to offer, of course, are benchmarked against their own simulation methods and against their own experiments. And this is just an example of a very simple proof of principle case in which we use variants of algorithms that are well known for near-term quantum computers in order to locate the conical intersections. So this is an example of a molecule that is very simple. So we use so the first study is really as a proof of principle, and however we can demonstrate that this can be done. Okay, and now the question is how to scale it, and of course the scaling will happen by combining the three main framework that I described before. So compilation algorithms that allow to minimize the depth of the circuits, so to condense the calculations, if you want, of the simulations in a very short circuits that is compatible, of course, with what can be done today and will be able to be run into tomorrow and quantum computers, and at the same time find efficient ways of extracting information by means of informationally complete framework. At the end, we always have the noise mitigation that removes the errors. And in particular, our noise mitigation uses methods that are tensor networks for which there exists already very efficient HPC approaches. So to summarize, and this goes a little bit also, allows me to go a bit towards the business model here. This is the type of complexity that one needs to tackle in order to really provide or work and move towards utility with quantum computers. So we have a layer in which we use these informationally complete measurement approaches in order to really minimize the number of readouts or really statistical errors, if you want, that is performed when measuring observables, and therefore when measuring for extracting information, like, for example, binding energies. Then we need to have techniques. Again, these are techniques that we developed based on informationally complete measurements in order to generate in a variational manner the state of the molecule mapped into the quantum computer and adapted in order to find, for example, ground state energies and excited state energy that are needed to understand processes like photodynamics. And then a very important piece is the mapping of fermionic problems, electronic structure problems, into qubits. And also this can be optimized, and it can also be optimized depending on the type of quantum computer that is used. And finally, a bunch of algorithms. These are just few of them, but there are very many that needs to be used in order to give, in a way, to find the best combination of medicinal chemistry techniques that are currently used and the quantum techniques that are available now today, and again, in a hybrid framework. And finally, going upwards, the network medicine, that is the place where we give context to the cell biology. Now, what is unique of what we do is certainly at the quantum simulation layer and the network medicine layer. The quantum chemistry layer is something where there are, of course, we have our own algorithms, but there are many alternatives. What makes it different and what makes it scalable is the fact that we can calculate, using informationally complete data, we can calculate many properties at once of the chemistry problem by using, and therefore making it more scalable, to use simple terms. So the main business model used by Algorithmiq is very similar to what innovation platforms, classical innovation platforms using, for example, machine learning, are doing in the drug discovery space. So it's partnerships based on the analysis of certain problems and use cases that are provided by the pharma, CRO, or whoever client. And then, of course, this is, as usually is done, divided into milestones with an upfront payment, and then subsequent payments at the delivery of when reaching out the existing milestones. We, of course, also work in a different, at a different level, at the level of the primitives, in terms of licensing. So, and this is again conversations with hardware providers who are interested in adding up to their offering, so quantum hardware offering, primitives for extracting information and for canceling noise that allow to work in the direction of more qubits, more complex circuits, so many more gates towards quantum utility. So I think I will probably end here, and I hope that I will. I gave you a panoramic view. I didn't go into many details, but I think that I will be very happy to answer to your questions